Good morning, good afternoon, and good evening, everyone, and welcome to this Inside Scientific webinar titled Developing Prefrontal Circuits and Their Role in Health and Disease. I'm Andrew Davies from the events team here at scientist.com, and I'll be your host today. This webinar has been sponsored by Neurotar, so a big thank you to them for helping make this event possible. We're very fortunate to be joined by Dr. Ileana Hanganu-Opatz, Director of the Institute for Developmental Neurophysiology at the University Medical Center Hamburg-Eppendorf, as well as Dr. Jastyn Pöpplau and Marilena Hnida from her group. They will be discussing their research on prefrontal networks in developing head-fixed mice and the role of these networks in cognition and models of neurodevelopmental diseases. And with that, I'd like to welcome our first presenter, Ileana Hanganu-Opatz. Thank you so much for joining us today, Ileana. The floor is yours whenever you're ready.
Yeah, hello everybody. My name is Ileana Hanganu-Opatz and I'm the head of the Institute of Developmental Neurophysiology here in Hamburg at the University Medical Center. It is a great pleasure for me to be here today and to introduce the topic on developing prefrontal circuits and their role in health and disease. It is a great pleasure, and I'm very happy that we have this webinar. Many thanks to Neurotar and Inside Scientific for putting this together. And I will briefly introduce the topic of my group. My lab is interested on brain development, and we are mainly interested on the brain activity. And it is, I think, for everybody well known that the brain generates pattern of oscillatory activities that have been described since long time. I think Hans Berger, more than 100 years ago, described them for the first time and recorded them electroencephalographically. This oscillatory activity of the adult brain is not only a correlate of a specific behavioral state, but it is also a very efficient neural code allowing different representation. And this brain activity in the adult has been well investigated. However, we still don't know what's going on during development. The activity at early stages of development has been recorded too. And interestingly, it has a different pattern. You can see it already by eye that in contrast to these continuous discharges that are present here, during development, the brain activity is discontinuous, meaning that periods of silence, almost like a flat line, alternates with these discharges. And with age, these periods of silence are shorter and shorter, as depicted here for a child, and then turns out to be continuous later in life. Now, I think it is obvious for ethical and technical reasons, it is very difficult to understand what's going on, what are the mechanisms behind these patterns of electrical activity during early stages of development. And it's also very difficult to know whether this oscillatory activity, it is relevant for the abilities that the brain has later on. To understand this or to address these questions, one strategy is to investigate animal models. And we are quite lucky because the animal models that are mostly used in a lab, the rats and the mice, are born at a very immature stage of brain development. They are so-called altricious species, meaning that brain development, for example, corresponding to the period that I showed you before with this discontinuous patterns of oscillatory activity, the second and the third gestational trimester in humans corresponds to the first and second postnatal weeks in a rat or a mouse, as depicted here. And indeed, when looking at the oscillatory discharges that are present in between these periods of silence depicted here for the primary somatosensory and primary visual cortex in the rodent, when you look at the single discharges in humans and rodents, it is obvious that they are very, very similar. They have an amplitude that increases and decreases, and the frequency switches between different bands, slower or faster. These patterns of oscillatory activity have been mainly investigated in the sensory systems, due to the fact that they are very easy to address then using different stimuli. And we learned a lot about the role of early oscillations for the maturation of the sensory system. And I think one of the pioneering findings has been obtained by my former colleagues in Marseille that could show that these oscillatory discharges in the sensory system, in this particular case in the somatosensory system, are important for the maturation of the tactile abilities that rodents do with this big whiskers that correspond somehow to our tactile, our touch perception. However, we and also the rodents are not only able to touch or to see, but we have also much more complex abilities, such as attention, problem solving, strategies development, recognition, switching, all kinds of memory forms, short or long term, as well as salience detection. And for all these abilities, there is a big circuit or circuitries all over the brain involved, certainly. But there is one brain area that acts as a kind of hub, and this is a prefrontal cortex that has been investigated in humans and in rodents. The prefrontal cortex, as depicted here, is built of several subdivisions, and it's involved in all the abilities that are depicted here. My lab is interested on the development of this high-order cognitive abilities, and we aim to address the general question whether adult cognitive abilities rely on the electrical activity of the developing brain. We have a specific focus on the prefrontal cortex, but of course, we consider all the circuits that are centered on this prefrontal cortex and include other cortical as well as subcortical areas. For addressing this question, we use a whole battery of techniques. You will hear about some of them in this today webinar. Electrophysiology, mainly in vivo and in vitro, combined with behavioral investigation and immunohistochemistry, optogenetics, and all other techniques that are necessary for monitoring as well as manipulating the activity in these developing circuits. This question is a big question. So it's a question that cannot be addressed immediately. And as you imagine, it composes different aspects, and we would like to focus on this aspect. And therefore, we somehow divided the work in several packages over the years. And during the first 10 years of my lab, we were mainly focusing on the very early activity corresponding to the time window that I presented to you in 2 slides ago, with these discontinuous discharges that corresponds to the first, second postnatal week in mouse and rat. But currently, we switch a bit the focus, and we would like to address as one of the first questions, which are the mechanisms that control the late development. And we are talking here about late childhood and adolescent period of cortical circuits and how they account for cognitive processing. So what are the mechanisms of this oscillatory activity and what are the cortical circuits involved? We already published some data that support the critical role of prefrontal circuits and activity for the later cognitive processing. And when I'm saying we, it's always a team, and this team is called Juvenile Development. It is led by Jastyn Pöpplau, and together with the others addresses this question. And you will have the opportunity today, after this short intro talk that I will give, to listen to Jastyn and Marilena that will present the data corresponding to this first question. Moreover, we are interested on the processing mechanism. So how do developing circuits compute internal and external information to set the cortical subcortical wiring? This means that we would like to understand the circuitry, the mechanism, the way how neurons communicate with each other, how they build functional ensembles, how these ensembles communicate with each other for the first time when the animal is performing a specific task. The team Neuronal Processing is led by Mattia Cini, and together with Irina Pokinok, Tony Mann, and Henrik Ösby, they address this question. The whole investigation of the normal development, it is nothing else than the control experiment for an important question that concerns disease. Which dysfunction within developing circuits lead to cognitive impairment and neuropsychiatric disorders such as schizophrenia and autism, and how can they be rescued? Because I think this is an important question, whether we can find a kind of signatures of disease that might represent amenable targets for therapies. And this question is addressed by Anne Günther, that is a team leader of the team Mental Disease. And she worked together with Malte Stelzer and Rebecca Kringel. And we already published the substantial data showing the relevance of this prefrontal activity for schizophrenia animal models. By the studies were led by Shasha Shu and by Lyndon Song. And finally, of course, is the prefrontal cortex the core or the hub of this cognitive circuits, but there is not isolated. It receives a substantial input from the sensory areas. As a matter of fact, it's a prefrontal cortex, a multisensory area par excellence. The question that I would like to address is how do early sensory inputs shape the cortical circuits accounting for cognitive processing? And the team Cognitive Olfaction, led by Sebastian Bitzenhofer and composed by Yunan Chen, Johanna Koska, and Fiona Pabst, address this question with a specific focus on olfaction. Now we will move on, and I would like to hand over to Jastyn Pöpplau that will present you the very recent data on that address this question: which mechanisms control the late development of cortical circuits accounting for cognitive processing? Thank you very much, and we are looking forward to your questions after the webinar. Thank you.
Yeah, thank you very much, Opatz, for this very kind introduction. Yeah, first of all, I would also like to thank the organizers for this great opportunity to give the presentation today. Today, I will talk about mechanisms that drive functional development, functional prefrontal development. For this, I would like to take you on a short journey by starting with projects that address these very early postnatal stages in mice, which was also, like Professor Hanganu-Opatz mentioned, the main focus of our institute during the last years. And then I would like to go over to more recent data where we extended our investigations towards the adolescent period. But before I show you this data or the data of my presentation, I would like to spend a few words on why it's so important to investigate the development of the prefrontal cortex. So the prefrontal cortex is implicated in a wide range of cognitive functions, ranging from the top-down control of attention up to working memory. While some of these functions, such as speech production or abstract reasoning, are thought to be inherently human, other functions can be also found in mice, since all mammals have common needs and need to adapt their behavior accordingly. This is a very important notion for us, since we are using mice to investigate the developmental mechanisms of such complex functions. While a clear structural homologue to the human prefrontal cortex is missing here in rodents, the brain areas that underlie these functional homologues can be investigated to gain a better understanding of the cellular mechanisms that mediate cognitive functions. Most of these cognitive functions develop quite late in life, so even after the emergence and fine-tuning of sensory motor abilities, cognitive functions still continue to improve. And this is thought to be caused by the delayed structural and functional development of the prefrontal cortex, which reaches its full maturity not before the end of the adolescence. At adult age, activity patterns have been found to highly correlate to cognitive functioning, but also to dysfunctioning. For example, here in humans, it has been described that prefrontal activity, especially within gamma frequency ranges, increases with task difficulty, but also with working memory load. On the other hand, in patients that suffer from schizophrenia, these dynamics are absent. And similar findings have been found in mouse models of mental disorders during cognitive tasks. This relationship between cognitive performance and prefrontal activity has been well described for adult age. However, how they mature and change across development to enable later these functions is poorly understood. Thereby, it's clearly visible at these example traces of mice at different ages that there occurs a prominent change across development. So as Professor Hanganu-Opatz already mentioned, from discontinuous burst of activity to continuous activity with higher magnitude. Given the sparsity of early activity, we asked in the first step if there are features of early activity that remain throughout development, and if these features also have an impact on adult prefrontal functioning. But before we are able to address these questions, we first had to tackle a methodological issue, because previously we did recordings in neonatal mice under urethane anesthesia. So to extend now our investigations towards the whole developmental period, we had first to address how anesthesia affects developing brain activity to rule out confounding effects. So we found that in neonatal mice, when activity is still discontinuous, anesthesia increases the proportion of these silent periods, but without affecting the spectral structure of these active periods. So consequently, when we only analyze these active periods, we found almost no anesthesia-induced changes. On the other hand, in juvenile mice, so when activity is continuous, we found that anesthesia increases the power of slow delta frequencies, but decreases strongly the power of faster frequencies. Therefore, we had to move to the establishment to non-anesthetized head-fixed recordings. And for this, the mobile home cage setup from Neurota was really a big help for this, because even lightweight and young mice can move this light air-floated cage really easy. Marilena Hnida will later also tell you more details about our surgery and recording protocol. So equipped with this new methodology, we were then able to answer our questions that we asked. And for this, we started targeting different cellular subpopulations of the prefrontal cortex with an optogenetic tool to investigate their impact on developing brain activity. This revealed that especially layer 2-3 pyramidal neurons in the prefrontal cortex are able already at early postnatal stages to induce rhythmic oscillatory activity. This layer 2-3 induced activity increases in its frequency and amplitude across development by starting at around 16 Hz at early postnatal stages until reaching classical gamma frequencies during juvenile age. This layer 2-3 induced activity was quite similar to changes that we observed during spontaneous activity and indicates that layer 2-3 pyramidal neuron might contribute to the development of gamma oscillation. And it also suggests that they might have already a functional importance at early postnatal stages, which might also impact the later development of the prefrontal cortex. This was confirmed by investigating early activity in a mouse model of mental illness. So this dual-hit GE mice, for genetic and environmental, carry a truncated form of the developmental risk gene disrupted in schizophrenia 1 and receive additionally a tail injection with poly-IC to mimic a viral infection during pregnancy. So we then recorded these mice at 8 to 10 days of age with a 4-shunt electrode that covers all layers of the prefrontal cortex to enable a layer-specific comparison of network activity. This comparison revealed that the power of spike-triggered oscillations as well as the firing rate of neurons was very similar between control and GE mice in layer 5-6. On the other hand, these parameters were strongly reduced for layer 2-3. We were able to identify a similar picture on morphological level. So the dendritic complexity and spine density of layer 5-6 neurons was very similar between the groups. But layer 2-3 pyramidal neurons showed a strong reduction in the dendritic complexity and also in their spine density. These neuronal alterations in GE mice were also accompanied by a hypermajor microglia network, which also most likely contributed to the observed deficits. Therefore, we attempt in the next step to rescue these alterations by adding minocycline, which is a commonly used microglia inhibitor, to the drinking water of the dam. during the first postnatal week. And this treatment was able to rescue the functional, morphological, and also behavioral deficits during a recency recognition task in GE mice. On the other hand, when we applied the same treatment protocol to a later time point, GE mice still showed behavioral alterations. This indicates a really time window specific sensitivity of early prefrontal activity for later development. But to prove this, we decided in the next step to chronically manipulate early activity. For this, we focused again on layer 2-3 pyramidal neurons according to our previous results. We found that chronic transcranial stimulation of this population from P7 to 11 led to a temporal augmentation of dendritic complexity. However, this was normalized at an older age. On the other hand, the strengths of prefrontal gamma oscillation as well as behavioral abilities were lastingly affected and were strongly reduced. So the synchrony and also the strength of gamma oscillation was reduced, and on behavior level, the early stimulated mice showed a strong reduction in their working memory abilities. But when we applied the same stimulation protocol 5 days later, we only found minor differences between early stimulated mice and controls. These data are also in line with the data of the GE mice and suggest really a time window specific sensitivity and also contribution of early activity for later functional development of the prefrontal cortex. Now I would like to shortly summarize this first part that I showed you, where we investigated 2 different mouse models, and we found that altered early activity due to a shared dysfunctionality of layer 2-3 pyramidal neurons really results in a lasting alteration of prefrontal functioning. And this shows that early activity is predictive for adult behavioral disabilities, but also, which our investigations of the ES mice showed, is not only predictive, but also actively contributes to a functional maturation of the prefrontal cortex. However, the question remains if prefrontal activity has an impact on adult functioning beyond the neonatal period. Because of the late maturation of major cognitive abilities, it is most likely that further time windows of sensitivity exist for the prefrontal cortex. Therefore, I would like to now go over to more recent data where we tried to identify functional and structural changes that occur in the adolescent prefrontal cortex. And in the next step, by interfering with identified processes, we aim to identify if the adolescent period is still a sensitive period for functional prefrontal development. For this, we recorded mice chronically from P16 until young adult age, while mice head fixed and allowed to move or rest, and recordings were performed in layer 2-3 of the prefrontal cortex. As you can see here from these example traces of mice at different ages, as well as in this 3D plot, which summarizes all recordings over age, prefrontal activity doesn't simply increase after postnatal development. First, prefrontal activity increases until early adolescence, then it decreases, and then it increases again to reach adult levels of activity. To quantify this, we focused on slow and fast frequencies separately. For frequencies above 12 Hz, we detected the frequency where the most prominent amplitude occurs. This showed that peak frequencies increase in their frequency across development from around 30 Hz at P16 until around 65 Hz at adult age. Peak amplitudes increased as well, but peaked during early adolescence, decreased afterwards, and increased again. This nonlinear dynamic of prefrontal gamma oscillations was also confirmed with a cubic spline model, which was able to cover the data better than a linear regression model. On the other hand, slow frequencies below 12 Hz didn't change significantly with age, neither in their peak frequency nor in their amplitude. However, the peaks, which are mainly confined to the theta frequency range, got better defined with age. In the early ages, we only detected one main peak at around 5 Hz, but with age, 4 Hz and 8 Hz peak became more prominent. To investigate the possible cellular substrate of this nonlinear reorganization of prefrontal gamma oscillations, we focus on the next step of prefrontal spiking activity. For this, we distinguished, according to the waveform shape, fast spiking units, which mainly correspond to parvalbumin-expressing interneurons, from regular spiking neurons, which contribute to our layer 2-3 pyramidal neurons. This clustering showed that firing rates of regular spiking neurons peaked during adolescence, and also their spike interval showed the lowest value during early adolescence. On the other hand, fast spiking neurons didn't change their firing patterns across development and only showed a slight narrowing of the interspike interval with age. Also correlated activity between regular spiking neurons, so the layer 2-3 pyramidal neurons, peaked during early adolescence, decreased afterwards, and increased again during adult age, and there also showed more focused patterns within prelimbic and infralimbic brain regions. Because this analysis was carried out on very short time scale, it suggests that early adolescent neurons show increased functional connectivity. And resemble also with this the dynamics we found for gamma oscillations. In the next step, we were interested if these functional changes in the prefrontal cortex, especially of layer 2-3 pyramidal neurons, also relate to the structure. Therefore, we target these specific neurons with a fluorophore by using in utero electroporation, and then we quantified the dendritic complexity with Scholl analysis. This showed that the dendritic complexity as well as the spine density of early adolescent neurons peaked. These structural changes across adolescence might at least partially be mediated by microglia cells. And therefore, we had a look in the next step at the morphology of prefrontal microglia cells. And we found that microglia cells of adolescent mice showed the least rounded morphology, which indicates higher phagocytic activity. To prove this, we also quantified postsynaptic densities of layer 2-3 pyramidal neurons, which lie within lysosomes of microglia cells, so which would correspond here to the pink overlay. This showed that the volume and also the number of inclusions were increased for adolescent microglia cells. These data suggest that microglia cells contribute to this structural but also functional remodeling of the prefrontal cortex during adolescence. After identifying how the prefrontal cortex changes, the activity of the prefrontal cortex changes along adolescence, we aimed in the next step to interfere with these processes to investigate if they are mandatory for functional prefrontal development. And therefore, we used a PLX diet during the time period where we identified the functional and structural peak. Since the PLX diet affects the whole brain to ablate microglia, we also recorded from the somatosensory cortex as a cortical control area. Over the course of PLX treatment to ablate microglia from the brain, the number of microglia got strongly reduced, and also the surviving portion of cells showed a significantly more ramified morphology. On the other hand, 4 days after treatment termination, microglia cells already started to repopulate the brain and showed the morphological phenotype most closest to the controls. To investigate the consequences of this adolescent microglia ablation, we looked in the first step at prefrontal and somatosensory activity. This revealed prominent differences, so prefrontal theta power was significantly reduced, and gamma power was significantly increased and shifted also to faster frequency ranges. On single unit level, especially regular spiking neurons showed increased firing rates and also correlated activity, whereas fast spiking neurons were not affected. On the other hand, in the somatosensory cortex, we found no treatment-induced changes, neither on oscillatory level nor on spiking level. This suggests that especially the prefrontal cortex is vulnerable to adolescent microglia ablation. To prove if these changes in prefrontal activity also affect the behavioral abilities of the treated mice, we used in the next step a prefrontal-dependent spatial working memory task, where the mice had to remember over several days and trials in which arms they can find food. And as you can see here, these heat maps of control and treated mice, the controls remembered over the days where they can find food, whereas the treated mice showed more random arm visits. And also when quantifying this over the days, the control mice made less working memory and reference errors, whereas the treated mice improved less. These behavioral disabilities were also correlated to changes in their prefrontal activity on single mouse level. So in the end, I would like to shortly summarize the second part that I showed you. First, we used continuous monitoring of prefrontal activity and found that it doesn't simply increase after postnatal development, but rather follows a nonlinear reorganization. These changes in prefrontal activity were also accompanied by a structural peak in layer 2-3 pyramidal neuron dendritic complexity and also with in the phagocytic activity of microglia cells. In the next step, we interfered with normal adolescent development by ablating microglia from the brain and found that this results in lasting alterations of prefrontal functioning. And with this, these data indicate also the adolescent period as a sensitive period for cognitive development, with also a comparable sensitivity to the neonatal period. And with this, I would like to thank you for your attention, and I also would like to thank Professor Ileana Hanganu-Opatz and her whole institute. There I would like to thank especially Sebastian Bitzenhofer and Mattia Cini, because they were the major contributors to the first part that I showed you. Now Marilena Hnida will continue with her presentation, and she will focus not only on the prefrontal cortex, but also on the hippocampus and how the prefrontal hippocampal communication evolves along the juvenile period, and this not only during functional development, but also in models of disease. Thanks.
Thank you very much, Ileana and Jastyn, for those wonderful presentations. Marilena, take it away whenever you're ready.
Hello, everyone, and thank you, Jastyn, for the introduction and your great talk about the development of prefrontal cortex and its role in cognitive abilities. And as mentioned, an important input area is the hippocampus, and on this prefrontal hippocampal network during late development, my talk will focus. And as it's challenging to investigate mice during late development, I will give a short introduction to the background of my research and show some main results, but I will also put emphasis on our methodological approach. We consider late development in mice starting at around P14, when they start opening their eyes and leave the nest, start exploring the environment. And we continue our research until P60, when the mice reach fully sexual maturity and are early adults. This time period in mice loosely translates to childhood and puberty in humans. And during this adolescence, cognitive abilities increase, and they depend on the prefrontal cortex. The prefrontal cortex forms reciprocal connections with the hippocampus, and on this hippocampal inputs to the prefrontal cortex, functions such as working memory rely. And the basis for this network is already present in neonatal mice, as was shown earlier in our lab, that in neonatal mice, the prefrontal cortex and hippocampus oscillate synchronously, and the hippocampus already sends a big input to the prefrontal cortex. From here, the question arises, how does this network develop so that from this helpless neonates fully function in adult form, and what happens during mental disorders, such as we see this dysfunction in working memory. Mental disorders such as schizophrenia often are seen as in prodromal symptoms during adolescence, and also often is seen the first psychotic episode. The dysfunctions often seen, such as working memory, rely on this prefrontal hippocampal network, and it can be translated to mouse model of these diseases. And it was shown early in our lab that already in neonatal mice, the prefrontal hippocampal synchrony in the mouse model is disturbed, and also that pre-juvenile mice show dysfunctions in a recognition memory task that relies on the hippocampus prefrontal network. So we aim at investigating the developmental trajectory in a mouse model here that mimics the 22q11.2 deletion syndrome, which is a syndrome that is based on a genetic deletion on chromosome 22 of either 60 genes or 35 genes. And it shows symptoms such as craniofacial abnormalities, but also more severe ones such as cardiovascular and immune deficiencies that can go along with a reduced life expectancy. The syndrome has a strong bidirectional relationship with schizophrenia, as around 25 to 30% of patients develop schizophrenia, and this genetic mutation accounts for 1 to 2% of sporadic schizophrenia cases. Patients show also deficits in cognitive abilities and in sensory motor gating, working memory, and fear conditioning, which are also typically seen in other schizophrenia patients. This deletion syndrome can be modeled in mice, as the same sequence of genes can be deleted on chromosome 16. This DF16A plus-minus mouse model that we use was developed in the lab of Joseph Gogos at Columbia University in New York. And next to this construct validity due to the same genetic mutation, it shows a phase validity, as here shown by Kimberly Stark and colleagues, and that the mice also show sensory motor gating deficits, as seen in the pre-pulse inhibition, and in deficits in a fear conditioning due to this freezing, decreased freezing to a conditioned stimulus. In a study from Sigurdsson et al., there was shown that the mice also showed, adult mice also show a reduced working memory, and with this, in this working memory task, also a reduced coherence between prefrontal cortex and hippocampus. So our aim is investigating this prefrontal hippocampal network during late development, detect if there are possible vulnerable periods, and compare the results to this disease model. We want to achieve this by performing simultaneous electrophysiological recordings of prefrontal cortex and hippocampus in mice chronically during P16 up to P60. We want to use awake mice because anesthesia was shown that it affects electrophysiological activity during development differently. And we use an invasive method as we want to record local field potential and multi-unit activity. So we either need to implant an electrode chronically, or we need to insert the electrode acutely for every recording, but for this, the mice need to be head fixed. We decided for the second method because it yielded the most stable results so far. And we use, for the head fixation system, we use the mobile home cage device from Neurotar, which is a platform that has holes where pressurized air comes through and lift a carbon cage that the mice can move freely, so it mimics the freely movement in mice. And investigating the same mice over this adolescent period, is challenging as the mice grow from around 8 gram at P14 to around 25 gram at P60, so they triple in weight. Also in pre-juvenile mice, the skull is still very thin and soft, and the mice are still in maternal care. So how we face these challenges, I will go through our experimental protocol. We do the surgery at P14 under isoflurane anesthesia and with buprenorphine as an analgesia. We fix the mice in a stereotactic frame, but not with the ear bars into the ears as they are still too small, but we use blunt ends to fix it with a very little pressure on the side of the skull. And we prepare it by removing the skin and cleaning it. And we mark our recording sites as we do not perform the craniotomy right away, but only after the training. And we use 3D printed custom-made windows that we glue around the recording sites so that we prevent dental cement from flowing over. We implant a silver wire as a reference electrode above the cerebellum. Then when we apply the windows, we apply one first layer of dental cement. Then we use this head plates from Neurotar that we then fix with dental cement, as we can't use screws for fixing the plates as the skull is still too soft. And here you can see a pre-juvenile mouse right after the surgery. So we have different types of head plates for different recording sites. And we can also use multiple windows so we can reach different sites in the brain. And it is stable, the implant is stable from the pre-juvenile mouse that we implant until up to the early adult mouse with P60. After the surgery, we let the mice wake up. It takes, with the isoflurane anesthesia, about 30 to 45 minutes. And then we place them back to the mother, and we additionally feed the mother with high caloric milk, which we prepare either from a lactose-free milk powder or we use a lactose-free milk for cats. Then we let the mice recover for one night and start with the training the next day. We always handle our mice very well, which is also pretty easy in the young mice as they get used to humans more easily than adults. Then we let the mice freely explore the carbon cage and the head fixation system. And when they are showing behavior that they are not interested anymore and want to move out, we start the head fix training. Our first tries we did with just the carbon cage, but it seemed too slippery for the young mice, so we added a Depron mat. And as you can see in the video, the young mice can move the carbon cage where they want to go. After the training, we do the craniotomy, and then on the next day we can start our recordings. We use 16-channel silicon probes, either 100 spaced for prefrontal cortex or 50 spaced for the hippocampus. We insert them acutely with an electrical arm, so we can go in very slowly and very on the same speed. And we retract the electrode in the same way. And we leave at least 1 to 2 days between the recordings, all this to minimize the damage to the tissue. And as you can see in the videos, we went to a lower carbon cage so that we can track the pupil dilation at the same time and have another readout for the activity state of the animal. We monitor the weight across development, and we can compare the weight of implanted mouse to not manipulated. Here it's shown for every recording, and the weight does not differ between the two conditions, so neither implanted and not implanted. The trajectory is different, and also in the DF16 mice, the weight increase is the same as in the wild type animals. When we record, as you can see in the scheme with the two, we have the two electrodes. The mice, we also track movement. We use this neurotar software that comes with the mobile home cage. And you can see a reconstructed heat map from two mice, that how they moved around in the mobile home cage. And we use it also to look at the average speed, ratio of movement, and distance traveled. And we can see that across age, there's no difference, as well as between the two condition groups. To align the movement to the electrophysiological recording, we send a TTL to our amplifier so that we can then align the electrophysiological recording exactly to when the mouse is moving or resting, and then we can look at both activity states. Here you can see examples of the recordings in different age groups. So P16, P30, 40, and 60, each exemplary for one recording in one mouse. And the reconstructed recording sites. In pink is the dye staining, which we perform after the last recording. And we cover the electrode in dye and insert it one more time to stain the recording site. We record in the prelimbic area of the prefrontal cortex and in the intermediate CA1 of the hippocampus. And we look at local field potential, and we filter it to 1 to 100 Hertz, and at multi-unit activity, which you can see in the lower traces, and filtered for 500 to 5000 Hertz, and this multi-unit activity can divide it into single units by a clustering algorithm that is waveform based. Here are the results for hippocampus activity. Here I look at the power, so the local field activity potential. And here's the power left are the power spectrum during resting and moving periods for 1 to 100 Hertz. And yellow to blue for wild type animals, and orange to red for DF16 animals, each with an increasing intensity with the increasing age. You can see for hippocampal theta activity, so 2 to 12 Hertz, that there's no change either in age nor in the two condition groups. But in the gamma activity, we have an increase across age. And during moving, we have this higher theta power, but also here there's no difference between the two condition groups. This picture is different when we look at prefrontal activity. Also here we don't have a change in theta activity, but in this mouse model, in the DF16 mice, we don't see this nonlinear increase in gamma power, but the gamma power is already higher in pre-juvenile animals, and this is the same way across the whole development. So we have a higher local activity in the prefrontal cortex across the whole developmental period. And there we can ask, does this come from disturbed input from hippocampus? Therefore, we look at the synchrony between the two areas, and for the spectrum, we look at the imaginary coherence, which looks at coherence without volume conduction, so without the instantaneous activity of the brain. And the synchrony between the two areas is highest in the theta range, and it is decreasing in the DF16 model from early adolescent to early adulthood, while this is not the case in the wild type animals. And during movement, when it's usually higher in adult mice, we also see that it's in early adult mice already disturbed in the DF16 mice, but not earlier on in the development. Imaginary coherence, however, is no measurement for directionality. So we also look at the normalized spectral density ratio, which gives an information about where the input comes from. And you can see also here during activity, the hippocampal input to the PFC is lower in early adulthood in the DF16 mice. Now, to sum this up, we have in the hippocampus an increase in the local activity during adolescence. Next to this, already described nonlinear increase in gamma activity in the prefrontal cortex, while the synchrony between the two areas does not change. But in the DF16 model, we have already early on a disturbed prefrontal activity with this higher gamma power, although we do not see a difference in the hippocampus. And the synchrony is only later on disturbed, not starting with the pre-juvenile period, but only in the early adults. Here we just look at the freely moving behavior of the animals, but we know that in adolescence also the social interaction peak. And this relies on the prefrontal cortex. Therefore, we plan on performing this as well. And this test was already established earlier in the lab and can be performed with the mobile home cage setup, with this maze from Neurotar, which has two chambers where one can place a mouse or an object. And the mouse that is recorded is in the inner big, can move around in the inner big part, so it can move itself towards one of the two chambers. We can align, we can use the movement, the tracker software to gain a heat map of where the mouse spends most time, as you can see in the right lower corner. In the video, you can see how the mice can, and the other picture, you can see how the mice can interact while they are in this maze. Here, this social preference task. So if the mice prefer to interact with another mouse over an object, was tested in this early stimulation model, which is this models an increased prefrontal activity in this vulnerable time period of P7 to 11. And we see a decreased discrimination ratio of mouse versus object. So the early stimulated mice spend less time with the mouse than with the object and with the mouse. Also, if you compare no interaction time with interaction time with the mouse, the ES show less interaction with the mouse. And comparing no interaction versus interaction with the object, there's no difference. By sending a TTL when the mouse is in either the area where the other mouse is or in the area where the object is, we can, to the amplifier, we can align electrophysiological activity again to the behavioral output. And we see that in the prefrontal activity, the gamma power is higher in the control mice during the interaction with the mouse than in the early stimulated mice. From here, I would like to come to a short second conclusion about our methodological approaches. We can, with the mobile home cage and our surgery protocol and recording protocol, we can monitor electrophysiological activity in mice, in the same mice across development from P16 to P60 in several brain areas, and align this activity to movement or resting periods, and also perform behaviors, innate behavioral tasks like the social preference task. With this, I would like to thank the whole lab for support and contribution to this work, and Ileana Hanganu-Opatz for give me the opportunity to perform my research in her lab, the organizers, Inside Scientific and Neurotar for this webinar, for the organization of this webinar, and of course you for your attention. Thank you.
Thank you, Ileana, Jastyn, and Marilena for those fantastic presentations. It is now my pleasure to welcome Jastyn and Marilena back for the Q&A session. And welcome back, and let's jump right in. So our first question here Do you think that similar developmental mechanisms to what you've presented here are present in humans? And if that's the case, are there any studies that support this?
That is a good question. Marilena, it's fine. I would start, and I think you also have something to say to it. Okay, yeah, so what first comes to my mind, the second part where I presented something about the adolescent period and what structural changes we find during this period. As I know, there are also many postmortem studies in humans showing that there's a temporal augmentation of gray matter and also brain volume and also spine density that peak during adolescence, especially within frontal regions. And yeah, that really nicely also mirrors what we see in the mice. And also there are some studies showing that stimulus-induced activity during working memory is altered during adolescence and is increased in comparison to younger or older age. So it really, nicely fits to what we are seeing in the mouse. And I'm also looking forward. We now also would like to extend this to more cognitive behavioral tasks to see if the changes during behavior also match the age-dependent changes in humans. So,
fantastic.
Yeah, and looking at the neuropsychiatric diseases, it's already known that in prodromal symptoms, they appear already during adolescence. So we would expect a similar development in the prefrontal cortex hippocampal network. And a nice aim would be that finding periods or finding markers that could be used to see in patients or see before patients have the first onset and already could have a window where you could start treatment earlier.
Great. So would one possible outcome of this work then be to identify patients before the development of symptoms, for example?
So that would be basically, that would be a great solution if you could do this. And there are indeed already during early activity markers that are also seen before the onset of symptoms. For example, there is a quite nice, which you also can measure with EEG. There is this what we call 1/f slope. So there's a tendency, if you calculate the power spectra of the neuronal activity, there's this tendency that with increasing frequency, the power of oscillation decreases. And this leads to a steepness of the power spectra. And if you fit this with a linear regression model, you have the slope. And the slope can give you a lot of information about the excitation inhibition balance within the network. And we see across development a shift towards more inhibition within the prefrontal cortex. And in mouse models of mental disorders, this shift is altered. And interestingly, when we look at EEG data of humans, so we have some correlations with them, we also see a similar shift, age-dependent shift in the humans. So it might be that especially this readout might be a quite nice symptom. Yeah, it describes the symptoms quite nice, or could be an early biomarker of disease. So let's see what's coming out of our future collaborations. But this one is especially really promising.
Fantastic. Okay.
There would be one really nice outcome. Another one might be that we've identified time windows where the change, where there are especially changes within the two areas or the network in mental disorders, and where when relating this time windows to humans to maybe find periods where treatment could be most effective.
Fantastic. Okay, that's wonderful. A couple of methodological questions as well for you here. The first one is, does the chronic implantation and recording procedure impact brain development? And if yes, how could this influence your results?
That is really a good question. Of course, when we established this method, we did a lot of control experiments to account for such possible effects. And the first thing that we did, we compared the activity of chronically recorded mice to ones that were acutely recorded. And we see also with a lot of mice no changes. So they have pretty similar brain activity at adult age. So that's the first hint that it's not inducing major changes with the brain activity. However, we also looked at the morphology, and it's indeed the case that the motor areas are less curved, so that leads to a slightly lower cortical thickness of the motor areas. However, gross brain dimensions and also the brain size, as well as the cortical thickness of the areas that are recording from, is not affected. So that's pretty similar between controls and chronically recorded mice. And to rule out that if there are some effects, to rule them out, we also use for our statistics linear mixed effect models, where we, for example, also account for the recording number to rule out if there are effects, to account for them within our statistical model. So.
Okay, fantastic. So you've done a number of validation studies, and as far as you can tell, there is no impact effectively then. Wonderful. Another sort of methodological question. Your time for acclimating the mice to the head fixed condition is quite short. Why are you using such short training time, and is it sufficient, basically?
To the why, we use it because we do want to start the recordings as early as we can. So we try to monitor across the whole developmental period. And yes, it seems to work. It works very well. It's if you use the really young mice, they seem to be more flexible and to adapt to this change in how they can behave during this head fixation. They seem to adapt very well. And since we don't see any difference in movement during the whole recording session, so across the whole age frame, there's no difference, even though later on they were on the mobile home cage much more often. We don't see a difference. So they adapt very fast, and the time is sufficient to train them. We do the training at least 2 times for 30 minutes.
Okay, fantastic. So in effect, you're looking at a narrow developmental window. So you, out of necessity, then need to use relatively brief windows for acclimation, I suppose.
Yeah.
Yes.
Fantastic. Here's a great question, I think. Does oscillatory activity influence myelination patterns in developing neurons?
That is a good question. I have to admit that we never had a detailed look into it. So we only have some preliminary data. For example, in the early stimulation model, we looked at the corpus callosum, where the interhemispheric connections are connected, and there is especially this myelination is really mandatory to ensure good communication between the brain hemispheres. And we found indeed that this early stimulation leads also to an increase in myelination. So there are also other studies showing that indeed activity also influences myelination. So now it is known that in mental diseases, the myelination strength, especially within interhemispheric connectivity, is reduced, as also the communication between brain hemispheres. So we are now planning within another project also to look at the myelination strength within our mouse models and compare the two controls, because as we presented, many of them have lower or altered activity during development, which might also affect the myelination.
Okay, fantastic. Unfortunately, I think we're just about out of time, a little bit over time here. So I think maybe we'll leave off with one last question that is a bit speculative, perhaps. But many of the projects that you showed focused on the L2 and 3 pyramidal neurons. Why did you focus on those? And do you think similar dynamics are present in the other prefrontal layers?
The special layer to 3 pyramidal neurons, yeah. As I already showed you, so this population really, we started focusing on them because it was really the only population that we found at neonatal age that really induces fast rhythmic activity. So these patterns are very sparse during neonatal development. And then during the later studies, we also found that this population is specifically affected in this GE mouse model. And also when stimulating them, we really induce lasting changes of the prefrontal circuitry. And it is interestingly also known that in humans, for example, with schizophrenia, especially this layer 2-3 pyramidal neuron population is strongly affected. For example, the patients and postmortem studies showed that they have decreased spine densities or also an increased number of differentially expressed genes. And there are also studies showing, also coming back to this microglia-neuron interaction, that especially these layer 2-3 pyramidal neurons are vulnerable to microglia-mediated overpruning. So as we saw it in our model, the microglia are all everywhere, but only within layer 2-3, they have this dysregulation of normal homeostasis. And I think it's really a population where one should focus more on, because it's really many studies showing that also in other disease, especially this population is affected. And we try now to address that on more molecular level to somehow identify markers why these neurons are so sensitive to such processes.
Okay, fantastic. Unfortunately, I think we're out of time, but I'd really like to thank you guys, Jastyn, Marilena, and thank you as well to Ileana for having you with us today and sharing your insights. It's been an absolute pleasure.
For us too. Thanks.
Thanks very much. A big thank you also to everyone in the audience for participating. And last but not least, we'd like to thank Neurotar for making this event possible. In closing, we hope you enjoyed this Inside Scientific webinar, and we hope to see you again next time. Thank you.